everyone. Thanks for the organizations and invitations from MVIF. And it's really my honor to share our research with you. Today, my topic is about the data-driven predictions of colonization outcomes for complex microbial communities. I'm Lu from Shenzhen, China. We all know that microbial communities played fundamental roles in our planet. It involved the health and disease for both the plants, the soil, the animals, and the host. And colonization resistance as emergent properties of the communities. It was a function in maintaining the compositions and functions of the community. Take the human gut microbiota, for example. It was constantly challenged with pathogens or probiotics we take and other living microbial drugs we want to take. But the invaders, they can colonize or not in the communities. This is the questions we want to address in this research. But when it comes to predictions of the colonization outcomes in complex microbial communities, we may have several challenges. The first is also take gut microbiota, for example. The human gut microbiota will know that it's highly personalized and dynamic system. It's nearly impossible for us to isolate all the species and do the pairwise experiment to build the ecological network. Besides that, the commensals, they may have different mechanisms to interact with the invaders. So without this background knowledge, can we use a data-driven approach to do the colonization outcome predictions? So we first want to validate this hypothesis in synthetical data. Let's consider we have a species pool with n species in it, and many local communities that are different in the compositions. They are subset species of the species pool. And when the invaders come, these communities can both permissive or resistant to the invaders. So it comes to different outcomes. That means the steady state abundance of the invaders is different. So data-driven approach means if we can collect enough data. Data approach means if we collect enough data about the community structures as input and colonization outcome as output. So by machine learning approach, we may build a map from the community structures to the colonization outcome. And we can do the prediction test. So we synthesize the data by Java models, and we found that machine learning models, they can solve both the classification test and also the regression test with good performance. But we need to pay attention that the sample size is quite important if we want to get good performance. We recommend that the sample size should be twofold of the species number in the community. After that, in silico data, we want to further validate the predictions in the real data. And we choose to use human stool-derived in vitro communities to do the invasion experiments. And as I just said, the sample size is important enough to get enough samples. We collect 24 stool samples from different subjects and treat them with 12 antibiotics. So in summary, we get nearly 300 communities with different compositions. After the real dilutions in vitro, we introduce the invaders here, and the real dilution again till the community come to a steady state, and we collect the samples here. We use the metagenomic sequence to get the baseline community structures and also the post invasion state abundance. In this experiment, we choose the two representative species as invaders. The first one is Enterococcus faecium, which was frequently reported as pathogens such as VRE. And we found that E. faecium only colonized in a small proportions of the community, as you can see in the PKI plot, such as random forest can predict if E. faecium can colonize in the community or not. And as for Akkermansia, it's a famous next generation prebiotics. We can say in the PKI plot, the communities are more permissive to it. It colonizes in most of the communities and with wide range the abundance. And machine learning models also have a good performance in predict the regression. That means it can predict the steady state abundance of the Akkermansias in the community. So the third part, we want to further use the data-driven approach to help us try to understand the ecology of the microbial communities. Random forest can infer the species that have strongest colonization impact on the invasions or faecium. And we found that faecalis was expected to be the strongest impact species. And we further validate the interactions in pairwise experiment, and we find that faecalis can directly inhibit the growth of faecium. Furthermore, we use the in vitro community culture method to test if faecalis can modulate the colonization in the community. We find that once we introduce faecalis into the baseline, even with different background compositions, faecalis can significantly inhibit the invasions of faecium. Okay, at last, I will give a brief summaries about the data-driven predictions of colonization outcome for complex microbial communities. First, we did the simulations and verify the prediction and make sure that the sample size is quite important. And the second part is we use the real data to validate the predictions of colonization outcomes with two different species. The third part is we use the data-driven approach to open the black box of predictions and try to understand the interactions, the species interactions, and modulate the functions of the community. And at last, thanks for your time and listening, and thanks for all the members in our lab, and the supervisors, Dr. Lei Zai and Yang Yuliu. And at last, many thanks to my collaborators, Xuwen Wang. He is responsible for the prediction part in this research. And if you have any questions, welcome to the QA sections tomorrow, maybe. Thanks again.